All right. Well, good morning, everyone. I'm Christine, one of the PGY2s, and I have a case that I saw actually my intern year on the internal medicine service, and neurology was consulted. So we have a 71-year-old male. He came in with 4 days of vertigo, nystagmus, and unsteady gait. Past medical history was not significant. His social history, he had a pretty significant social smoking history as well as alcohol history. He was a very outdoorsy person, was a hunter. When he got to the ED, his vital signs were unremarkable. He didn't have a fever. His initial exam, he was awake and alert. His speech was appropriate and fluent. He was completely neurointact except for this spontaneous bilateral torsional nystagmus that did not change with direction. And he wasn't able to walk anymore. And this was a pretty significant decline from his baseline. His labs were unremarkable. Given the concern for possible posterior circulation stroke, he had an MRI brain that was negative. And then due to his inability to walk, he had total spine imaging, which was also negative for any kind of high-grade stenosis or cord compression. He was actually admitted to the medicine service for additional workup and therapy evaluations. And during his next couple of hospital days, had a pretty significant change in his exam with progressive worsening of his mental status and cognitive changes. At that point, neurology was consulted. He became non-responsive to any kind of verbal or aggressive stimulation. He was really only groaning from pain. He had these erratic multi-directional eye movements of variable amplitude that were more consistent actually with opsoclonus than nystagmus, as well as increased tone in his upper extremities and his neck bilaterally. He was hyporeflexic and had downgoing toes. We talked a little bit more to his wife, and she was saying that he actually had around 2 to 3 weeks of low-grade fevers before he came in, as well as some headaches and some unusual behaviors. It sounds like he was repeating all the words that she was saying back to her, was having hallucinations and kind of moving items around the house that weren't supposed to be in those places. Asked a little bit more about his hunting history, and it sounds like he skins all the deers that he hunts and may have cut himself with his hunting knife, and was also bit by a squirrel a couple of weeks ago, but didn't seek care at that time. We also asked her about weight loss, and she said he had around 10 to 15 pound weight loss in the past 3 months. At that point, we were concerned for possible infectious cause, like a meningitis, and so we initiated broad CNS coverage plus doxycycline and acyclovir. Put him on EEG, which just showed generalized mild diffuse slowing without any seizure activity, and got an LP, had a cell count of 13 white cells with an 81% lymphocytic predominance. His protein glucose was normal. He had 6 oligoclonal bands, and his meningitis panel on PCR was negative, and culture was negative too. We sent off kind of more fancy encephalopathy panels, but those were all still pending, and sent off some other infectious and inflammatory and nutritional workup based on his social history, and those were all negative too. The next day, he had worsening decline in his GCS. He was more tachypnic. There was concern that he wasn't protecting his airway, and so he was transferred to the neuro ICU. With additional concern for a possible, inflammatory encephalitis, he was started on empiric Solumedrol 1 gram per day. Got a repeat brain MRI, this time with contrast, and again, that was completely normal, unrevealing. With his smoking history and concern for a possible paraneoplastic syndrome, we got a CT chest, abdomen, and pelvis that showed a right middle lobe mass that was concerning for malignancy. So initiated Plex, and he got 2 sessions of that while we went and scheduled his biopsy of his lung, which confirmed a small cell lung cancer. So finally got the results of those serum and CSF panel, and he had positive ANA1 or anti-WHO antibodies in both his serum and his CNF. And in the serum, it was a high titer positive. So we diagnosed him with subacute paraneoplastic syndrome with opsoclonus and cerebellar degeneration with encephalopathy. A little bit more about the ANA1 or anti-WHO antibody. The antigen itself is actually in the family of RNA binding proteins, both found in the peripheral and central nervous system. It's most associated with small cell lung cancer, like in our case, but also with neuroblastoma in children. CSF often shows inflammation in 90% of cases, like in ours, and while the MRI brain is often normal. And the neurosymptoms are broad. You can have a subacute sensory neuronopathy, which our patient did not have, but also this limbic encephalitis, kind of strange behavior changes, and the cerebellar degeneration, like he was having difficulty ambulating, and the nystagmus, as well as this opsoclonus myoclonus syndrome. And I just wanted to talk about how the titers do matter. You can have these antibodies detected at low levels in serum with the small cell lung cancer, but you don't always have to have neurologic symptoms with those low titers, and that's a good prognostic factor, whereas the high titer correlates with neurologic symptoms. So in these cases, unfortunately, neuro outcomes are poor. In 75% of the cases, folks remain bedridden once the disease has plateaued, and improvement is rare even with anti-tumor therapy and immunosuppressive therapy. The median survival is around 7 to 13 months with an overall poor quality of life, and the death either comes from the tumor pathology itself or from the neurologic complications. So for our patient's outcome, talked to the oncology team, and there were no kind of available chemotherapeutic options for his lung cancer. And then just based on his very quick progressive decline in functional status and quality of life, goals of care conversations were had, and there was withdrawal of care and patient death.
Thank you, Christine.
Yeah, you're welcome.
And Andy, I'm just going to take the screen for 10 seconds because apparently I was not sharing my screen, and I just wanted to share the picture of Ernie crying because he loved the number 2 so much. And the great photos that came in from Suma Shah, those are her and Kevin's 2 children, and Roshni and friends during Diwali. So Andy, back to you.
Well, thanks, Rich. I'd like to say that was my first egregious lack of knowledge of protocol that for this week, but sadly that would be a lie. So
and Will is away this week too, so we didn't have the assistant grand rounds person.
I blame Will. Yes, that's a good point. And Clemens, I mentioned you were from Austria. I think that might not be right. Didn't you mention there's an area just south of Liechtenstein? Is that right?
Well, yes. So there is a little German-speaking tribe in the middle of the Alps, located in Italy, and that's where I'm from.
And what's the name of the?
It's a state of Italy called South Tyrol.
South Tyrol.
Yeah.
Very good. That's interesting. It sounds very mountainous.
It is. It's only mountains, Dolomites, and steep valleys.
So I don't want to take up any more time. I think, just to say as we get on to your recent work, I think what you've done with biomarkers and some of these risk models has in really no small way shaped the way NIH and Michael J. Fox is moving forward to build out these databases and discoveries to help figure out how they can positively affect management and treatment of these diseases. And so I think we're all looking forward to hearing about it. So thank you for joining us this morning.
Great. Thank you so much, Andy, for the kind introduction. Thank you, Richard, for inviting me, and happy Diwali, and hi to everybody. It's great to be here. So I'm looking forward to give you sort of an overview of the work we've been doing to make baby steps towards developing a predictive and hopefully precise neurology. Parkinson's is our key disease. There are 7 million of Parkinson's patients today in the world, but it's the fastest growing brain disease, actually outpacing Alzheimer's disease, and forecast to reach 14 million patients in 2040. And the costs, the health burden on patients, caregivers, and society is enormous, with 52 billion annually in the US alone. Today, we treat Parkinson's patients reactively using a one-size-fits-all approach. We wait until they present symptoms. We wait until the symptoms progress before we intervene. We're focused on clinical phenotypes, really sort of 20th and 19th century phenotypes, and we treat patients as if they were all the same. The focus is really on replacing the deficit in dopaminergic projections in Parkinson's patients. The DAT scan was FDA approved a few years ago. And if you scan a patient with Parkinson's disease, you don't have to, actually you can make the diagnosis clinically, but if you do, you will see in almost all cases a reduction in the dopamine transporter radionucleotide ligand, particularly in the putamen. And it's typically asymmetrical, so worse on one side than on the other compared to healthy controls. Now, if you would look at the midbrain of Parkinson's patients, this is where the dopaminergic projections are arising from the substantia nigra, which you can see here in a normal midbrain, where melanized dopamine neurons form these arborizations that go all the way up into the basal ganglia and the forebrain, and in some cases, reach to 2 or more meters of length in the neuritic projections. Now, a midbrain of Parkinson's patients, there's a massive loss of dopamine neurons, which even macroscopically is visible. And so we go down a laundry list of about 20 different types of dopamine replacement medications or deep brain stimulation to treat these patients. But in essence, we treat patients as if they were all the same. But let me introduce you very briefly to 2 of my patients to convince you that this might not be the case. One patient is John. He actually was an executive or is an executive at an insurance company and has had Parkinson's for more than 10 years, is doing extremely well, and he's enjoying life playing with his grandchildren. He recently retired, and his main concern in life is really to get in 165 rounds of golf per year. And this graph shows you the mini mental state exam scores over time in John and patients like John. Very slow decline on average over time. By contrast, Kevin is actually a molecular biologist, a PhD, also 10 years, more than 10 years into his Parkinson's disease. And within 7-8 years, his wife came home and found him staring into space, looking at things that were not there, grasping for things that were not there. He's now wheelchair bound. He has lost his memory. He's confused. And his wife, who is a nurse, had to quit her job and is now providing 24/7 care to Kevin. Now, Kevin's mini mental state exam and patients like Kevin's exam is shown in the magenta line with a much more dramatic and steeper decline over time. So what we're hoping to do is to shift healthcare of Parkinson's from a reactive to a predictive, preventive, and precise medicine. And if we are able to do that, it could have a big impact on patients. So how does this work? Well, we really need 2 components to achieve this. Number one, we need to be able to predict disease complications or rapid progressors. So we need to have predictive tools to tell who will be a fast progressor, who will have complications, who will be slow progressing. And number 2, we need interventions, medicines that prevent disease from progressing in specific patients. And if we succeed, we should be able to push patients down from the red line to the green line, and thereby making an enormous impact into the quality of health. Because here in the motor Parkinson's space, which is really a prodromal disease for Parkinson's disease, dementia, there's a lot we can do for the patients. Whereas here, there's, things get really tough. one could also push this even further away, and Andy and many others are working on this, trying to detect even the prodromal symptoms and preventing even motor PD from occurring. So how are we going about this? We have what I think of as a total information awareness approach. We are on a mission to map the Parkinson. We want to know all data in all patients at all times. We are particularly focused on mapping the genome, the transcriptome, to some degree the metabolome, and as well as clinical phenotypes in Parkinson's patients over time. And we use three main platforms. I will touch up on two today in the first part of the talk: working patients in deeply characterized patient longitudinal cohorts, and secondly, in human brain cells. And with this dense Parkinson engine, we're hoping to identify the disease driver in each patient, then go to our freezers, search in the CSF and blood samples for tailored biomarkers, identify compounds that can serve as precision medicines in collaboration with pharmaceutical companies or in-house, take stem cells from our freezers for preclinical trials in a dish, recontact the patients, and launch proof of concept, small scale precision trials. And so let me give you sort of some glimpses of how this is going. And it really, a major eye-opener for me was started with GBA, the beta-glucocerebrosidase gene. When we found that in our all-comers and Parkinson's patients just walking into the movement clinic at Brigham and Women's Hospital, Mass General Hospital, 10% of them turned out on sequencing, targeted sequencing of the GBA gene to have a protein coding mutation or variant in the GBA gene. So 10% of quote unquote idiopathic Parkinson's patients. And that's a huge number. And actually that makes GBA the most common well, let's call it monogenic form of the disease, although one could squibble with this. And now these GBA mutations, they come in different flavors, as shown here. Actually, a total of 350 mutations known, and loosely they can be grouped into severe mutations, mild mutations, and risk variants. And this grouping actually makes a lot of clinical sense because it's based on Gaucher's disease. And these in the Parkinson's patient are heterozygous carriers of GBA mutation. But in children with homozygous GBA mutations, a different disease arises in an autosomal recessive manner, which is Gaucher's disease. And this can either just involve the periphery, hepatosplenomegaly, as in this boy, or it can affect the CNS in severe mutation. And so the separation here we're showing with esophoria, strabismus, developmental abnormalities in children, in babies and infants. And so that's how we group GBA mutations into mild, no CNS involvement, and severe mutations. And this makes sense. This is important also for heterozygous Parkinson's patients, because when we look back at our graph, turns out that Kevin is actually carrying one of the severe GBA mutations, while John is GBA negative. And these intermediate lines are patients carrying mild, the green risk variants, and then mild and complex allele shown in brown and in orange. So another way to look at this is with a Kaplan-Meier survival curves. And so here now we're starting not with years in study, but disease onset, and follow the patients longitudinally over time. And what's really striking here is that based on these 4 types of GBA genotypes, we can identify 4 rates or 4 speeds of progression of Parkinson's disease that are quite dramatically different. So at 10 years, 21% of the non-carriers developed global cognitive impairment compared to 48% of patients carrying a severe Parkinson's, as a severe GBA mutation. And so there is this spectrum of clinically meaningful GBA mutations, and they lead to quantitative pathway changes. When we look at the enzyme encoded by the GBA gene, which is GCase, short GCase or beta-glucocerebrosidase, the activity was reduced in dried blood spots of Parkinson's patients, and this nicely quantitatively correlated with the severity of mutations. And similarly, both in plasma, which is shown here, as well as in CSF of these patients, there is a quantitative metabolic quantitative trait locus relationship with increased accumulation of this direct substrate glucosylceramide in plasma as well as CSF. So the mutation type regulates GCase activity, and that regulates substrate accumulation, which can be mapped and tracked and could be a biomarker of the disease pathway and of drug response. So this is really how we got excited about this and started about this. And so 10% of patients with a GBA mutation, that's really an important target, but it's a beginning. And we are really hoping to find the disease drivers in all Parkinson's patients. And to do this, we put together the IGIP consortium, the International Genetics of Parkinson Progression Consortium, that is going through different phases. Actually, everything I'm telling you about today is from phase 1, when we had about 4,000 patients that were longitudinally followed for 12 years with 31,000 visits. We're currently actually scaling this up to phase 2, where we will have more than 10,000 patients followed over time with deep phenotypes. And so what we did, what we're doing is to systematically scan the genome of these patients and using genome-wide survival studies to determine which loci, which variants regulate the speed of progression. We used whole genome genotyping in this analysis. So we imputed a total of 11 million SNPs, which is considerably more dense than previous Dziwis analysis. And also it's important conceptually that this is really a different way to do genetics, with lots of work being done over the last decades on Dziwis, which is case control association studies, searching for variants that distinguish cases and controls. But what we're doing is different. We're interested in longitudinal phenotypes and the critical time dimension, and that's why we're doing genome-wide survival studies, looking at cases over time. And what we found in the phase 1 of the IGIP analysis, which came out earlier this year in Nature Genetics, is 5 loci associated with cognitive progression of Parkinson's over time, specifically with the time to conversion from Parkinson's disease to Parkinson's disease dementia. And these are three novel loci identified with particularly strong signal in the RIMS2 locus, and also reaching genome-wide significance in TMEM108 and the VVOX locus. And so RIMS2 is a synaptic protein, and if we look at carriers with the RIMS2 variant versus non-carriers over time, there's a striking difference in the speed of progression. So the carriers are the interrupted magenta line, and now we are here looking at how many patients develop dementia, I'm sorry, over time. So these are all Parkinson's patients, but they're free of the dementia at disease onset. Everybody who already had dementia was excluded. And over time, more and more developed dementia. And so the RIMS2 carriers, 40% have developed dementia 10 years into the disease, compared to only 8% of the non-carriers. So it's a hazard ratio of 4.77. It's a very strong effect. However, it's a low frequency variant. So only 94 carriers were in this, out of these 3,800 patients. And similarly, in these patients, the RIMS2 carriers have a more rapid decline in Mini-Mental scores over time compared to the non-carriers. And so how does this compare to to GBA, which others and we previously identified as progression gene, and ApoE4, which is was confirmed in our analysis as a progression locus for progression from Parkinson's to Parkinson's disease dementia. And so RIMS2, so ApoE4 had a strong effect, and particularly the homozygote ApoE4 carriers, have a strongly accelerated cognitive decline. GBA, we talked about before. If you look at all mutations together, it was a twofold hazard ratio of 2 for developing PDD over time. But RIMS2 had a 2 to 3-fold stronger effect compared to ApoE and GBA. So what does RIMS2 do? Well, we're really just starting to look into this. It's really at the very beginning. There is some knowledge about the basic biology of RIMS2, thanks to Tom Südhoff's work at Stanford, who showed together with Pascal Kayser that the RIM family of proteins, of which RIMS2 is part, is involved in docking and priming synaptic vesicles for synaptic fusion. So they tether the synaptic vesicles to presynaptic calcium channels, and that's critical for 2 steps in the synaptic vesicle cycle, docking and priming, which are the steps immediately preceding vesicle fusion. And for the Parkinson's aficionados amongst you, so the fusion step is actually where synuclein is thought to be involved as a chaperone. So it's sort of intriguing link to this critical step for Parkinson's, and we're working on better understanding this. Now, instead of looking at the effect of one variant at a time, one can combine variant into polygenic scores. And this slide is actually is from Andy Singleton's work. It's the well, I guess I'm proud to say that this is the only slide that's not from our lab in this talk. And so they found 90 Dziwis peaks associated with risk of developing Parkinson's disease in healthy people. And if you combine this polygenic, this variance into polygenic score, patients in our Harvard biomarker study with in the highest tertile of the score have, you know, a 7.5 fold odds ratio, 7.5 fold increased risk of having Parkinson's disease compared to people not having the risk variant. So the polygenic susceptibility score looks promising for identifying healthy people at risk for Parkinson's disease. But can it help us in patients who come to our clinic? Because healthy people don't come to our clinics, right? It's patients who come to our clinics, and they are asking us to predict their future and to prevent bad things from happening. So can we use this polygenic susceptibility score to predict progression in patients? And the answer is no. Here, if we group Parkinson's patients into the lowest quartile of the polygenic susceptibility score versus the highest quartile, follow them over time, so there's no difference in their cognitive decline over time. By contrast, if we make a polygenic progression score that includes the 3 novel variants plus GBA and ApoE, carriers of this polygenic hazard score have a much more rapid progression over time compared to non-carriers. So it looks like it's actually really fascinating to me because it points at the divergence of the genetic architecture of progression and susceptibility. And it suggests that there are Parkinson's variants that are chiefly linked to susceptibility, and Parkinson's variants that are chiefly linked to progression. In addition, there is a third set of variants that regulate both susceptibility and progression. So this actually, you know, it's both interesting to me really from a mechanistic standpoint, but it's also really important for drug development, because depending on, you know, what you want your drug to do, this will suggest different drug targets. Now, genetics is critically important to identify disease drivers, and it gives us precise drug targets. But for the purpose of prediction, clinical data points are also highly informative, actually in many cases more informative. And so we are both scaling up the search for genetic drivers of progression, as well as clinical drivers using a phenome-wide approach. And this is sort of just the very beginning of this that we published in Lancet Neurology a few years ago, where we built a clinical and genetic predictor using the GBA gene, as well as clinical features which were machine learned from a small set of previously nominated candidate clinical predictors of the disease. And so by combining these 6 clinical features together with the GBA gene, we can actually quite accurately predict what patient will develop dementia over time with good accuracy. Actually, to be precise, so this graph here, we were predicting global cognitive impairment with 86% accuracy in this area under the curve. And in the replication cohort, which was completely independent longitudinal patient, it was similar. And this composite clinical genetic predictor was actually quite stable with the 10,000 random resampling iterations where we randomly reassembled a training and a test set, 10,000 times, and rebuilt the classifier, retested it in each of the 10,000 iterations. And so here these graphs show the areas under the curve observed during these 10,000 iterations. And so for prediction of dementia, this was quite stable with an area under the curve in the testing set on average of 87% accuracy, which is pretty good. So, all right, and similar to the polygenic score, one can use this clinical genetic scores to group patients into low scorers and high scorers, and then see how they do over time, as shown in this Kaplan-Meier curves, with very clear differences in the two groups. And actually, so in the high scorers, 73% now had cognitive impairment at 10 years. But importantly, the low scorers, they were virtually free of cognitive impairment at 10 years. So only 4% among the low scorers had memory issues. And in terms of applications, it's important. Hopefully, it will become important for the practice of medicine. But more immediately, it might be useful for clinical trials, because if you enroll rapid progressors into clinical trials, you increase your power to detect a positive drug effect if it exists. So if you have rapid progressors and your drug works, you'll be better able and quicker able to see this in a homogeneous group of rapid progressors. And we ran a power calculation on this. So in a traditional clinical trial, phase 3 clinical trial of all comers with Parkinson's disease, that last 2 years, you would need 1,600 patients to see an effect of an experimental drug on MoCA scores, compared to only 274 patients would be needed if only rapid progressors with a high clinical genetic score are enrolled. So, and we implemented this as a simple online calculator that's on the website, available for everybody. And so, with going forward with this, we're hoping that genetic predictors, either single genes or polygenic scores or clinical genetic scores, can be used to enrich a target population, stratify a clinical trial, and then with precision drugs be used to turn rapid progressors into slow progressors. And we also developed a biobank around this, the Harvard Biomarker Study Biobank. This samples are available for everybody. And I think we contributed about 10,000 samples over the last years to investigators around the world. And the corresponding data are available also openly through the AMPD Accelerating Medicines data portal. So everything we talked about so far was really about learning from patients and using genetics and clinical data to predict and prevent progression in patients. In the second part of the talk, I'm going to talk about this brain cells, our second platform, and more specifically, the question, how can we go from Dziwis variants to actually to drug targets in specific brain cells? And that's important because most of the Dziwis variants are non-coding, and they don't necessarily tell us what's the implicated gene, how are the Dziwis variants functioning, and how do they perturb brain cells. And so the overarching goal of this Parkinson's Cell Atlas, or PD5D, is to come up with a map, a wiring diagram that tells us how different genetic variants change gene expression and cellular phenotypes in specific brain cells in humans with Parkinson's disease. And we're using two premier technologies, or two major chief technologies. One is laser capture RNA-seq, which we developed, and the other one is multi-omics, particularly single nucleus and spatial transcriptomics. And well, I'll tell you about the really surprising and amazing finding from our laser capture RNA-seq work, and sort of give you a snapshot of what we're currently working on with multi-omics, single nucleus RNA-seq. And so how does this LC-RNA-seq work? Well, with laser capture under the microscope, we microdissect dopamine neurons or pyramidal neurons from autopsy brains of Parkinson's patients, healthy controls, extract the RNA, pool of 300 neurons per brain, and then very deeply sequence the RNA content. And the important thing here is that we are ultra deeply sequencing the RNA content, and that we're sequencing the entire transcriptome, not just polyadenylated mRNAs, RNAs, so not just mRNAs, but all RNAs. And we looked at, we captured dopamine neurons from 89 brains, pyramidal neurons now from 141 brains, as well as some control cells. And total of 80,000 neurons were laser captured, thanks to Jis Young and David Gritch in the lab, and very deeply sequenced. And so what did we find? Well, I guess first I want to convince you that this is really specific, that we're really specifically capturing dopamine neurons. Here, looking at neurofilament medium chain, which is a pan-neuronal marker, right? And if you look at pyramidal neurons or dopamine neurons, this neurofilament medium chain exon is highly transcribed. But if you look at a specific dopamine marker, dopamine receptor D2, there's zero transcription in the pyramidal neurons of these various exons from the gene model shown here, but a very nice transcription in the various dopamine neuron RNA-seq experiments. Okay, so what did we find? Well, to set this up right, really pretty much everything we know about the human brain is based on 1.2% of the human genome that encodes proteins. So 1.2% of the human genome encodes proteins, and that's really all we know about. Turns out in our LC-RNA-seq analysis, 64% of the human genome was actively transcribed in human brain dopamine neurons. So what number, including with transcripts from intronic, intergenic sequences, in addition to exonic sequences. So this, opens up really a vast, unexplored universe of non-coding RNAs. And actually, it's highly consistent with what the ENCODE consortium found in peripheral cell lines, where they also used total RNA sequencing and found that 62% of the human genome was actively transcribed in these cultured cells as well. So what do these non-coding RNAs do? Well, so so in our human dopamine neurons, we find the mRNAs about from 18,000 genes. We found long non-coding RNAs, about 8,000 on average. And in addition, we found 71,000 transcribed non-coding elements that we termed teeny. So these were novel, unknown transcribed elements, typically about 200 to 400 base pairs long. And we accumulated substantial evidence. It's far from definitive, but we accumulated substantial correlative evidence suggesting that a large part of these transcribed non-coding elements, or teeny, are enhancer RNAs. And so what are enhancer RNAs and enhancers? Just to make sure we're all on the same page. So everybody knows about the gene promoter that regulates how a target gene is transcribed. But turns out that chromosomes are not just linear, but they form three-dimensional loops where in some cases far distant regions of the chromosome are looped back to the promoter, form this enhancer complex, and regulate target gene transcription together with the promoter. And this is likely, well, the promoter is sort of a universal switch for gene expression. The enhancers fine-tune expression in specific cell types in response to disease and in environment. And when an enhancer is active in a neuron, enhancer RNA is transcribed actually in a bimodal direction, in both reverse and forward strand. And so this gives you this typical bimodal RNA-seq piles. And here for an enhancer RNA transcribed from an intron in the synuclein gene, where the alpha-synuclein protein in Lewy bodies accumulates in Parkinson's disease. So there is this active enhancer in intron 4 that's highly transcribed in dopamine neurons, and where 161 transcription factors, form a hotspot and occupy this region based on the ENCODE consortium work, and these active enhancers are supported by epigenetic features. So they also have the histone marks, H3K27 acetylation of active enhancers. So using total RNA sequencing, we can identify this putative enhancer RNAs that line up with epigenetic features of enhancers. And these novel enhancers actually really can drive gene expression in. So we didn't test to really prove it. You have to test all 71,000, but we tested a small subset, about 20 of these enhancers in neuronal cells and HeLa cells, and they indeed drive gene expression in cells, as well as in zebrafish, interestingly, where they were specifically driving expression in these neuronal clusters in the brain of zebrafish, as well as in the midbrain of mice, based on work of the VISTA consortium. So some of these enhancers are active, and they're also cell type specific, which really fits with the idea that they fine-tune gene expression in a cell type specific manner. So some are specific to dopamine neurons, some are specific to pyramidal neurons, and some are found in non-neuronal cells. And these patterns can be used to cluster dopamine versus pyramidal and non-neuronal cells. But so how does this help us to understand how genetic variants regulate cause disease and function in brain cells? And to make headway towards this important question, we overlaid Dziwis variants for any disease with the active enhancers, the 71,000 active enhancers we found in human brain dopamine neurons. And so it turns out that Dziwis traits, variants for Dziwis traits and diseases were highly enriched, highly overlocalized in enhancers actively transcribed in dopamine neurons, much more so than in exons or promoters. And by contrast, is in random, no enrichment in random genomic background. And so what disease variants were overlocalized to enhancers active in substantia nigra dopamine neurons? Excitingly, many of them are linked to dopamine system traits. And importantly for our work, Parkinson's variants, Dziwis variants were highly overlocalized to enhancers active in dopamine neurons. But not just Parkinson's variants, but also variants linked to phenotypes that are affected in Parkinson's patients. Sleep prominent, sleep related variants were also localizing to enhancers active in dopamine neurons, as well as heart rate variability traits. The Parkinson's patients lose heart rate variability and develop prominent sleep disorders, particularly REM sleep behavior disorder. So, but it actually started to get even more interesting because it wasn't just the hypodopaminergic traits localizing to dopamine neuron enhancers, but also hyperdopaminergic traits such as schizophrenia were very significantly overlocalized to active enhancers in dopamine neurons, both schizophrenia and variants linked to drug response in schizophrenia. And so this sort of suggests that that genetic variants might perturb the function of active enhancers in dopamine neurons and thereby change gene expression specifically in these neurons. And we have until 9 or...
Well, I think some of the clinicians may need to log off. I got a message from Rich, to sort of keep going, and those who can stay on can stay on.
I will wrap it up quickly. So basically, so this supports the idea that Dziwis variants function for gene regulatory effects, and this actually can be used for therapeutics. Some drugs can modulate enhancer activity, for example, beta-2 adrenoreceptor agonists, and thereby regulate expression of important genes, alpha-synuclein, for example. And now we're fortunate to have this ASAP consortium where we're taking this from laser capture RNA-seq to multi-omics single nucleus RNA-seq, and Duke, Andy just told me, actually got 2 of this 9 million awards. So congratulations to you guys. It's really amazing. What we are doing in our consortium is we're sequencing 1 million brain cells, and we're building a map that links genetic variants to expression changes in specific cell types. We see about 20 different types of brain cells in the midbrain and the temporal cortex of these 100 brains we're looking at. And we're also, we want to deconstruct and reconstruct the function in these brain cells in a spatially resolved manner. So we're also doing spatial transcriptomics and using this information to localize each cell type into the right position in space. And so this is actually coming along really nicely. This is H&E staining of a human temporal cortex. It's hard to see the layers. Here's the white matter, but using spatial transcriptomics and machine learning, actually you can uncover the 6 cortical layers and place the various cell types in the right position and see how this changes with different input genetic variants and during disease progression. And so to put this all together, we're working on single nucleus EQTL analysis, which will causally link genetic variants to expression changes in specific cell types. And that really brings me to the end of this talk. So we took a whirlwind tour first in patients, longitudinal cohorts, trying to use genetics and clinical data to develop predictors of progression. And in the second part, looking at brain cells and trying to wire genetic variants to gene expression changes in specific brain cells to find targets for precision drugs. So thank you very much for having me. And also I want to say thanks to my amazing lab who really was working hard to accomplish this, and happy to take any questions.
Thank you. That was really great. I think if anyone wants to put them in chat, we can read them, or feel free to unmute.
So I'll ask a question. So thank you, Clemens. So, the problem with this data in the past has been the lack of reproducibility by different labs. How are you going about that type of, to make sure that it's not just one lab, one protocol that, gives the results you've seen?
So, yes, so replication is really the key, and really sample size is the key. So for the genetic studies, here in phase 1, we had 4,000 patients followed with 30,000 visits. So now we are scaling it up to 10,000 patients. So if the strong signals will survive, the weak signals will fall to the wayside. And it really is going to be critical, for more longitudinal cohorts to be developed to increase the sample size and to be able to replicate it in these findings in additional patients. And for the transcriptomics, it's really a similar story. So the field really has massively developed. So with going from gene expression microarrays to RNA sequencing. So number one, the technological precision is increased. And number two, also, now we're starting to have the sample sizes that are that lead to meaningful results. For example, with our Parkinson cell atlas, we're sequencing 1 million brain cells, and other groups, are doing similar things. So there will be replication data sets emerging. And also, you didn't talk about this, but, for blood gene expression biomarkers. So there's actually very beautiful, the field is maturing. So initially, nobody had these RNA samples, right? So nobody had the money to do this in a large scale. So it was, 10 samples, 100 samples. Now we and others looking at 1,000 blood samples with RNA sequencing. And so you're starting to get, robust data once you're looking at, thousands of patients.
Kyle, you had a question?
Yes. So you have all these, new predictors now, and, whether loose or strong associations with cognitive decline and progression. Was wondering how that changes, how you approach a patient with these markers. What are you telling them? since it's no longer one size fits all.
Right. So we're really sort of on a transition period, and to give you practical answers. So what's most actionable for patients is if you can offer. So there are many trials ongoing directed at GBA PD and LRRK2 PD. And so therefore it can be meaningful for your patients, right? If they're interested in clinical trials, that would be meaningful to tell your patients, say, if there's a new trial enrolling for LRRK2 therapeutics, GBA therapeutics, you should be genotyped you for GBA and LRRK2. That would have, an actionable impact. That is one scenario. In other scenario, I guess then there are other scenarios that are sort of more specific to the patient situation.
Clemens, I've got a question about the spatial work. So we've been doing spatial with the Visium platform and looking at AD pathology. So I was just wondering how you're getting on with the analysis of the expression profiling, what's your approach to looking at the proximity of the tissue to Lewy bodies and things like that. And then for you, what's the next step moving into the spatial work? Right. So, well, then we share the fun of, working on this really cutting edge method for which actually many tools are still lacking and analytic. So we are staining for Lewy bodies as well. So we do one cryosection where we do the Visium, and then an adjacent section where we do Lewy body staining. We have not gotten to combining the two analyses yet. What we are currently working, so what we just solved is to uncover the cortical layers, based on gene expression patterns and machine learning. So that we have solved, and really the current problem we're trying to solve is, which has not been done in the literature, is how to basically build a reference temporal cortex around our 100 samples, 100 Visium samples, because each temporal cortex looks very different, right? And so the integration across multiple brains, that's what we're currently trying to figure out. Because I think, just quickly, I think the digital pathology and marrying it with things like transcriptomic profiling is going to be really interesting going forward. And I was just chatting to Andy West in the background that, I'd love to talk to you more because we're developing in situ sequencing here as well, which, improves on the resolution. Oh, cool. Great.
Andy, do you want to bring us home here?
I have a question. Do I? So, one of the things that's just emerged in the last year, maybe the last few years, I suppose, is this concept of, body first Parkinsonism and then, brain first. And, I suppose if you create two groups, all cases would fall in those two groups, if there's such a thing. But I wonder if, if progression is so heavily affected by, that kind of heterogeneity in origins that it's muting some of the power that you might have. And I'm, and I wonder if there's a way to pick apart strata where, it might encompass differential origins. I know it's all, speculative, but maybe, yeah.
So how would you identify or group patients into body first versus brain first, based on data we have?
Yes, that's a great question. And I know there's a raging debate right now in movement disorders on the stratification of like these PPMI samples. And, one of the things is like the GI features. And I know Roger is on the line here, at least he was. Versus those that don't have those changes. I don't know. But we don't capture a lot of that data in the MDSU PDRS. I think there's one question about, you know, constipation in part 1. Maybe some of the clinicians would know better than me, but you know, and that's one of the theories as to different origins. But it does strike me that there's so many, you know, loci for susceptibility, and it seems there's so few for progression, you know, so.
Right. I mean, so that question is really, I think, in large part related to that the progression work really just started like 3 years ago, right? Whereas the susceptibility work has been going on for 2 decades. Yeah.
Right. Yeah. Well, it's very exciting. I think it's the right, you know, the direction is going to yield a lot of important things we haven't realized yet, as you mentioned. So good.
Yeah, so thank you for arranging this, Andy. Clemens, thank you for talking to us about your incredible work, and everyone else, have a safe day.
All right. Bye-bye.
Bye-bye.
Everybody, thank you.